Um, all right, so it gives me great pleasure now to present our first speaker, Professor Colleen Hanlon. So the title of her talk is The Frontal Pole, an emerging TMS treatment target for drug and alcohol use disorders. Colleen is a professor at Wake Forest University School of Medicine in North Carolina, United States, where she leads an electromagnetic therapeutics research program. She spent her career identifying neural circuit irregularities in patients with drug and alcohol use disorders and modulating these circuits with non-invasive brain stimulation. Recently, she also served as a senior author on the first consensus paper published by a group of over 70 scientists from over 10 different countries outlining the path forward for non-invasive therapeutic development for addiction. So really important work. So if you're ready, Colleen, I'll hand over to you to get started.
Okay, thank you so much. Great. Can you all see my screen? Yes. Okay, excellent. Well, thank you all so much for inviting me to be a part of this. Make no mistake, I would way rather be in Melbourne right now. But maybe I'll take you up on an in-person invitation sometime in the future. But for now, today, I'm going to tell you a little about what we've been doing in terms of using TMS as an emerging therapeutic tool for addiction. And as Aaron said, I'm now at Wake Forest University School of Medicine. And so just in case you are interested in knowing where that is, we're in North Carolina, which you can see here on the red dot in the US. It's often known, the corridor from Winston-Salem, which is where I am, to Durham is often known as Tobacco Road. So we're really right down the street in broad terms from University of North Carolina and from Duke. So basketball is kind of in everybody's blood here. This is sometimes called Tobacco Road, and Tobacco Road is because all of these towns got kind of famous and their economies were driven by cigarettes. Winston-Salem is famous for Camel cigarettes. Reynolds Tobacco is headquartered here. And actually, while that is not necessarily something to be proud of, what is very cool is that over the last 10 years, the town has made a concerted effort to turn all of those old tobacco factories into a huge innovation quarter. And so we're here. And it's a great place to be. So I encourage you all, if you're ever happen to be in the general area, let me know, and I'd love to sort of be your hostess. And then this is just a disclosure slide. I've served as a consultant on various NIH grants. All the funding associated with the studies I'm going to present today was provided by the National Institute of Health. Okay, so our lab, even though we tend to study addiction, what I would like to say is we study something much more fundamental about human behavior. And that has been said by Rene Descartes and Kant, is this idea that there's a duality in the human experience, that we're driven by both emotion and by reason. So this is a mannerist painting here of the temptation of Adam. And as you probably know, the story goes, Adam and Eve are in the Garden of Eden, this beautiful thing, and they're promised an eternal life of happiness in Eden if they just don't do one thing, and that's to eat from this tree. And so here we have Adam, and he's faced with this shiny, beautiful red apple being given to him by this beautiful, seductive woman, and he has a choice to make. Does he take a bite out of that immediate rewarding reinforcer, or does he choose to live his life in this place of happiness? So we all kind of know how that turns out. And I like to tell that story because it's probably one of the oldest stories, at least of Western civilization. And it's because it's sort of this fundamental part of being human. Everyday modern struggles between this appetitive drive and cognitive control are present everywhere. So you or people you know may be driving down the highway, and maybe you have your phone in your passenger seat, and you may see a text message pop up, and for a split second, if you're like most people, you'll have an impulse to be like, ooh, what's that? And try to orient your attention to it. But hopefully you realize that you shouldn't actually look at that text message. You should keep your eyes on the road. And so this is a decision that's made. In much more common parlance, right? Many people around the world socially drink. And many of us have been in a scenario where we're out at a bar with friends. Perhaps if I was there with you in Australia, we would do this evening, and people would be hanging out and perhaps socially drinking. The evening would get late. Some people would start going home, and someone in the crowd might say like, oh, come on, one more round. You might think to yourself, oh, maybe I should stay and have another. That would be socially reinforcing. Maybe it would be free. Maybe it would taste good. That's this appetitive drive. But then hopefully you also think, but I have to drive home. I have a talk to give tomorrow. My significant other will be mad at me. And so you kind of weigh these two things. And for many of us, sometimes we go towards the appetitive drive, but hopefully most of the time, cognitive control comes into play. One of my favorite pictures is this one here of a Dunkin' Donuts, which right here puts this choice in your face. And it says, here, look at you. Do you imagine amazing smell of donuts coming at you as you walk down the street? And then they say 6 donuts for only $3. What a deal. And so that's your temptation. And then they put the choice right out there in front of you and they say, for only 900 to 2,800 calories. And so you make a choice here. So this is very common. And these choices are driven by neural circuits in the brain. We know a lot about some of the neural circuits that navigate emotion or arousal, cognition, and action. One way to think about these is the mesolimbic, mesocortical, and nigrostriatal dopamine systems. Although dopamine tends to be concentrated deep in the basal ganglia receives input from various cortical areas. And in this case, we have these frontal striatal thalamic circuits. The mesolimbic circuit, kind of the red circuit in this little cartoon, guides kind of hot cognition, vigilance, arousal, your drive to have that next drink. The mesocortical system regulates executive processing. Sometimes this is called cold cognition and reason, and your ability to evaluate the alternative reinforcers in your environment and guide yourself away from the immediate potent reinforcer. And then finally, an area which I think we often overlook in addiction research is the nigrostriatal system, which is motor control. It's really hard to have a substance use problem if you don't actually put the substance into your mouth or inject it into your arm, right? And so in many ways, for the ritual around drug use is intimately tied with your sensory motor system. And if we look at some sort of track tracing studies that have been done in non-human primates, we know that, again, these frontal cortical areas are directly related in an organized fashion to terminal areas in the basal ganglia. So when we think about this and these cortical striatal systems, we think, well, one way we could think about decreasing drug use behavior might be to increase executive control. So in this way, when I'm at the bar tonight and I'm tempted to have another drink, you increase my ability to say, no, you don't need to do that. You need to do these other things. Alternately, you could try to just decrease the reinforcing potency of the stimulus. So in this case, you could frankly just make the drink not taste as good. And this is actually a strategy that's being used a lot for alcohol. You can take a pill and it will decrease sort of the rewarding value of the alcoholic beverage that you drink. And then finally, another way that we think about with TMS, which I'm not going to talk about too much in this particular talk, is exploiting the motor system as a tool perhaps to promote opiate sparing among individuals that are using chronic opiates for pain. So you all are the Brain Stimulation Society. You're experts in all of this, but just to kind of review 2 key TMS principles that guide our decision-making, our conceptual model. The first one is stimulation depth. So we know that the strength of the stimulation, or the strength of the electric field, decays very quickly with distance. You can see here in a graph on the left side of your screen that sort of a decay rate of a standard figure-of-8 coil. And so one kind of general rule of thumb is that the amount of it's approximately about 2 centimeters from the surface of the coil at a given strength of the magnetic field, and you can actually depolarize neurons. So that's the strength in terms of the distance. There's also a spread, and the spread as well, if you just think of an electric field, it's kind of like a cone, the inverted cone that comes into the brain. And so there's some, as with depth, there's some width. And so as like a point of clarity for this, you'll often hear some coils, H coils, are described as deep TMS. And I suspect that I'm not alone on this seminar to say that we all have to remember that although those coils get deep, they also really get wide. So as deep as they go, they also get a lot of breadth. So a standard figure-of-8 coil, for example, I was talking to a reporter from National Geographic a few years ago, and she was in the TMS lab, and she wanted to know if you were to project the brain onto the globe, about how much territory is it stimulating? Is it like a country or a state or a city or a hemisphere? And so we kind of like, whoa, that's super cool. And so we went back to the lab and we sort of calculated approximately the surface area of the brain and the surface area of the globe, and the intensity or the size of the induced TMS electric field. And it turns out that a single pulse of TMS at about like 60% machine output activates an area about the size of India, which I actually think is sort of a fun little tidbit that we'll tell some of our participants sometimes, and they get a kick out of that. The second principle is transsynaptic modulation. So we know that although we're only directly modulating the cortex, because neuroscience works the way it does, if you activate a population of neurons enough, then they will summate in space and time and cause the next population of neurons down, the monosynaptic targets specifically, to also fire action potentials. And so in this, we know that this is true because when you place the TMS coil on the motor cortex, you get a contraction in your hand, right? And that's at least 2 neurons away. So we know that that's very true. And if we apply that philosophy to the prefrontal cortex, we know that we can stimulate the dorsolateral prefrontal cortex, or perhaps the medial prefrontal cortex, and also expect to get activation in the striatal terminals of those cortical afferents. So when a lot of what we know in TMS has been built on the shoulders of the early imagers in the field. Strafella in Toronto has done some outstanding work. George Etkin, Jess Daskalakis, Pasquale Leone, goes on and on and on of all this great strides that have been made to learn about this. We now know that dorsolateral prefrontal cortex is kind of the sort of standard target for treating depression, specifically the left, maybe the right. And I would argue that based upon what we know from our preclinical colleagues, that in fact the medial prefrontal cortex is probably a sort of translationally predicted target for treating substance use among our patients. And as Aaron mentioned before, there's actually a lot of people around the world currently now evaluating TMS and TDCS as potential treatment interventions for addiction. It wasn't that long ago that like 10 years ago, no, it was all depression research, and there were very, very few people studying addiction. And now there are so many people pursuing TMS as a treatment tool for addiction that we were able to bring together 77 authors to have some opinion or to weigh in on best practices moving forward. So there's a ton of momentum in the field, which is a great place to be. And then the world kind of changed or advanced in August of 2020 when BrainSway, who produces H coils, came out with the results for their first multi-site, double-blind, randomized, sham-controlled study in the US, at least. This is very US biased, I'm sorry. And what they showed, the FDA, it was kind of an FDA pivotal trial. And so the FDA cleared TMS, specifically delivered by the H4 coil, which kind of targets the insula. And I would like to say that actually generally targets all of the sort of lateral inferior prefrontal cortex down to the insula as a treatment for smoking cessation. So there was a lot of enthusiasm. I'm going to go through the details in just a second. But the take-home message is, you know, the results aren't really super compelling, but they're there. That efficacy is comparable to pharmacotherapy. And for us as brain stimulation researchers, this is great because there's been kind of a door that's opened to lead the way for many, many more people around the world to develop non-invasive brain stimulation treatments for addiction. And so a brief summary of what they did. They had 262 chronic smokers, 14 sites, double-blind, sham-controlled. Each participant received 15 sessions of active or sham TMS. That was approximately Monday through Friday, 3 times for 3 weeks. They then came back for several weeks and just received a single treatment like once a week, which I was told by the PI was actually had much more to do with just getting people to come back rather than an efficacy kind of rationale. And they use this coil, the H4 coil, which you can see here is the produced electric field map. It tends to be pretty lateral, bilateral on the left and the right. They seem to think that the site of action is the insula, which is because we do know there's some idea that individuals with an insula lesion or a lesion in the anterior insula tend to perhaps spontaneously stop smoking. This was a paper done about 15 years ago by Becerra and colleagues. So they measured the continuous quit rate after this, after these 18 sessions of TMS, or 15, the continuous quit rate from smoking. So it's a pretty high bar with at least the US FDA to get a treatment approved for smoking. You have to actually induce cessation. So just decreasing the number of cigarettes that you smoke is not considered a suitable dependent measure, which makes it pretty tough. Right? You smoke a pack of cigarettes a day and you go down to a quarter of a pack a day. That's not good enough currently in the eyes of the FDA. For alcohol, however, the endpoints are much easier to achieve. They've embraced sort of a harm reduction model for alcoholism. And so actually, I think that that is actually one of the reasons why we have more FDA approved interventions for alcoholism. And I hope, and I'm optimistic that TMS might be one sometime soon. But what they did in fact find is that there was an increase in the continuous quit rate amongst individuals that received real TMS versus sham TMS. And the efficacy was comparable to pharmacotherapeutics. So this is pretty good. I think, you know, it'll be interesting to see if it takes off as a treatment because it's obviously very expensive compared to the pharmacotherapeutics. But I think the optimistic angle of it is that we have a start, and now we can start to improve it, much like depression did, right? Depression started off with the standard protocol people were using, and now we know so much more about factors of TMS that optimize efficacy for depression and different pulse sequences, et cetera. So I'm hopeful that happens in the addiction space. Okay, so if we come back to our models, we could try to increase executive control to help people stop taking that extra drink. We could try to decrease cue reactivity, so just make that drink kind of less rewarding. Or we could somehow change the motor system to inhibit the action or to decouple the motor system with the prefrontal cortex system. So a few years ago now, there was some groundbreaking work that demonstrated in rodents that there's a causal relationship between stimulation of the ventral medial prefrontal cortex and drug-taking behavior. This was first shown in cocaine use behavior. So using optogenetics, we can decrease activity in the infralimbic cortex would cause, I believe, an increase in cocaine use activity, whereas if you decrease it in the prelimbic cortex, you actually go the other way. So what's really nice about that is it showed some causal relationship between the medial prefrontal cortex and drug-taking behavior. We looked at our own functional MRI studies. This is aggregated data from 156 individuals that had come through our lab over time. And all of these individuals went in the MRI scanner and received a functional MRI task where they were exposed to blocks of the drug cue or a neutral cue. And these people were either alcohol users, cocaine users, or heavy smokers. And what we found was that when you aggregate everyone together, the area most activated to drug cues versus neutral cues is the medial prefrontal cortex, which you can see on the image to the left, on that kind of big purple blob in the middle, as well as some anterior insula bilaterally. When you break that sample down by their primary drug of abuse, you find actually that the insula clusters are primarily driven by the cocaine users, whereas alcohol, nicotine, and cocaine users all have elevated activity in the medial prefrontal cortex. So again, this is kind of what the whole brain looks like. And so you really see a focusing here in kind of the medial prefrontal cortex. So we decided we would use that as our target for TMS treatment. There was good preclinical data. We had a lot of functional MRI data, which we used as like target identification data. So then we did an initial target engagement study. So this is kind of the way in which a lot of people are trying to do science these days, is target identification, target engagement, and then treatment trial. So before we invested a ton of time and effort in a multi-site randomized clinical trial, which is really slow, and you get a little scared, like what if you didn't pick the right protocol? We start off with these basic target engagement studies. And what we did here is we applied a single session, it's probably bad to say, but we applied 3,600 pulses of continuous theta burst stimulation to the left frontal pole. So we had to go left or right. Our data, actually, we did some reverse computational modeling of the peak hotspots in the brain, and that drove us to the midline FZ as our target. But we didn't actually want to go in the middle because, as you all probably know from remembering your neuroanatomy, there's a central sulcus, right? The poles come out and then they come back in. And so if we stimulated right in the middle, we were going to lose some of our juice, right? Because the TMS field decays very, very quickly with distance. So we had to go to the left or the right. And we decided to go to the left because the preponderance of evidence suggested that craving tended to be a little more common towards the left, medial prefrontal. So we stimulated 100 in the medial prefrontal cortex. So it's approximately here, the left frontal pole, at 110% of resting motor threshold. That is achievable. A lot of people are like, oh my gosh, that must hurt so much. But we have no problem with it whatsoever. The secret is in the slow ramping of the intensity, which I can talk about later if y'all have questions. And we ran it in 1,800 pulses, and then we stopped for 60 seconds, and then we did another 1,800 pulses. Because I know that you guys in Australia are some of the sort of industry leaders in novel and accelerated pulse sequences, I'll spend a little more time on this to tell you why we chose 3,600. So because for depression treatment, it was typically patients might receive 3,000 pulses. We wanted at the time to kind of keep this fairly stable. So we wanted to keep the pulse number about the same. Theta burst somehow comes in like 600 pulse units, just based on tradition, I guess. And we knew we were going to need to expend some of our energy with this ramping. So we took 3,000 and then we added another 600 pulses, which we were going to use for ramping. But it turns out that our machines at the time weren't capable of doing that and they would overheat. So we split it into two 1,800 pulse sections and we let the coil cool for 60 seconds in the middle. So those are the details. I'm just explaining that because I know several people on the call might be interested in that. I'm not sure that it's like the secret sauce. It's just something we chose. And it turns out that the data were pretty good. So where are we stimulating? Another thing that comes up is people, myself included, can be a little sloppy about what we call our brain regions. So we are stimulating the frontal pole. My friend and colleague Jonathan Downer has done a lot of studies where he is also stimulating the medial prefrontal cortex, but he in fact is stimulating a much more dorsal target, which from functional connectivity data you would suggest would have the inverse effect on behavior or craving as stimulating the ventral medial prefrontal cortex. You'll also hear a lot about these like connectome derived targets, which is very hot right now. And that's the dorsal lateral prefrontal cortex. Again, kind of different from the ventral medial. And then this in obsessive compulsive disorder, TMS has an indication using this coil called the H7 coil. And they are also stimulating the dorsal medial prefrontal cortex, but it's in truth is actually much further back from these other two targets. So I think as a field, we all really would do a service to one another to be very specific about our targeting. But just so you know, when someone says the medial prefrontal cortex, they could theoretically mean anywhere along this medial line. And there are, in fact, differences in the cytoarchitecture of this area we're stimulating, which is Brodmann area 10, compared to the motor cortex. So the motor cortex is agranular. It has a difference in thalamic inputs compared to the cortex, which is a really dense layer 4, et cetera. So there are differences which the reason to point that out is it may not in fact be the case that what we're learning about LTP-like and LTD-like protocols in the motor cortex may not generalize to these particular prefrontal cortex areas as conveniently as we might like. Okay, one more TMS principle before I get into the data. We also know, again, from a lot of work that's been done in Australia, that there seems to be state-dependent effects of TMS. So a lot of this has been done in the motor system, showing that if you prime the hand or you prime the individual, you can actually amplify the effects of TMS itself. And in fact, in many ways, something like theta burst might not have the same directionality of effect, depending upon if it was primed or unprimed to begin with. In psychiatry world, that also has been demonstrated with PTSD and with drug abuse. And so in all of our studies, we actually are priming the individuals about their drug use before they receive the TMS. And they're often, we're having them look at drug-related cues while they're receiving the treatment. Okay, so our first sort of target engagement study, here we took individuals, in this case 25 cocaine users and 25 alcohol users. We put them in the MRI scanner and we looked at their brain reactivity to drug cues versus non-drug cues. And what we found was that at baseline, the cocaine users had high functional connectivity between the medial prefrontal cortex, again, this is kind of ventral medial, and the putamen, which is the dorsal striatum. And the alcohol users also had high functional connectivity between the medial prefrontal cortex and many aspects of the striatum, including the caudate and the putamen and the insula. Everybody received a single session of real or sham TMS. I should go back. So in this case, it was, we call it a TMS sandwich design. So they came in, they got scanned, then they got tapped on, and then they got scanned again. And then they came back 2 weeks later, generally, they got scanned and they got tapped on with either the real or sham, whatever they didn't do before, and then they got scanned again. So everyone has 4 MRI scans. So you can imagine this is actually a very expensive study to do. And we compared the difference in the functional connectivity pre to post TMS when they received real versus when they received sham TMS. And what we found was that in the cocaine users, though, when you receive real TMS, there was a specific decrease in functional connectivity to cocaine cues between the medial prefrontal cortex, the left caudate and putamen, as well as the left insula. And in fact, the same was true in the alcohol users. The visit that they received real, there was a significant decrease following real TMS in connectivity between the medial prefrontal cortex and many of these same striatal regions. So that was very exciting. The idea that we showed this was transdiagnostically relevant also made us very excited and motivated us to move on to a big clinical trial. For full disclosure, we've done a lot of other things that haven't worked. As scientists, we always kind of say all the things that work, but in fact, most stuff doesn't work. So just kind of bearing that in mind, it's not like we have a secret sauce. We do a lot of things that don't work, that never get published and don't often get talked about, except for random interactions at a meeting. But this one worked, so I'm telling you about it. So we started a clinical trial. In this case, we had individuals that were coming in for intensive outpatient treatment for substance use. The way this is commonly done in the US is that insurance, so in this case, these were both covered by insurance. Insurance will tend to pay for about 30 sessions of counseling. And so there's these intensive outpatient treatment programs that arise where individuals will come in, they'll receive about 3 hours of group and individual counseling per day for 4 to 5 days per week for about 4 to 5 weeks. And so we had one actually at Medical University of South Carolina where I was before. And so we used their program. We took people that were coming into the program, and before they actually began their counseling, we looked at their brain reactivity to cues. And then while they were engaged in that multi-week intensive outpatient treatment program, before their therapy sessions, we gave them a single session of our CTBS 3600 pulse treatment directed to the left frontal pole. People were randomized to receive real or sham, and everyone received 10 sessions. So you could think of this as 10 days of TMS, but kind of like uber intense sessions. So 3,600 pulses is almost like 6 sessions in a day, but there's no time between the sessions themselves. They received 10 days of that. And then we had a 1-month follow-up and a 2-month follow-up where we did imaging and we looked at sobriety. This is just the basics of the enrollment. This is for the alcohol study. We screened about 100 people, 50 of them were randomized, about 25 to real and 25 to sham. And I don't have enough time to go through the data here. I think this is being recorded, so you're welcome to look at these anytime that you would like. And hopefully one of these days we will actually publish the study soon when I can stop procrastinating, and then they'll be out for everyone to see. But what I want to draw your attention to is that as circled in red here, our retention rate in the group that got real was much higher than the group that got sham. That was our first sign that things were going pretty well. In general, 84% of people completed all TMS sessions in both groups. So per this idea of feasibility, again, a lot of people think that it's not tolerable to get TMS to the frontal pole, but in fact, in our hands, we have no problems with it. So those are all good signs. This is the general demographics. And when you look down at the bottom, these are the primary clinical trial stats. And sort of, again, to kind of highlight the most interesting points, it looks like at approximately 3 months, so that's they initiate the study, they're in the intensive outpatient treatment for 1 month, there's a follow-up at 2 months and a follow-up at 3 months. The individuals that received sham TMS were 2 times as likely to relapse to alcohol as those that received real TMS. The other way to say that is that the likelihood of 3-month abstinence was 2 times as high in the group that received real TMS. So that's very encouraging. And Again, it's not a huge trial, of course, and this is only done at our site, but it's very positive. I think in particular, I'm most positive about TMS for alcohol compared to some of the other groups that we've studied. We also studied cocaine users. In this case, they were recruited from the VA system. So I don't know if you have a similar thing in Australia, but so there's basically a national health service in the US for veterans. And so if you're a veteran, you kind of get free healthcare for life. And so there's a system of hospitals around the US that take care of the veterans. And they really, actually, the mental health care through the VA system is outstanding. It's actually much better than is available than if I wanted to get it and I couldn't pay out of pocket. So we got these cocaine users from the VA hospital. So they tended to be a little more male, but their attention was very high. Oops. So if I draw your attention down to the bottom of the screen, the 3-month retention was identical in both the group that got real TMS and sham TMS. And we think that actually has to do with something unique about the veterans in the program. There have been several TMS studies that have been done in the VA system that have kind of odd results. And I've seen several presentations that explain why there's just kind of a group dynamic that sometimes is different in those individuals. But anyway, again, drawing your attention to feasibility, 76% of people in this case completed all TMS visits, which we thought was great. And if you look down to the clinical trial stats, in this case, the sobriety fell off in both groups. Cocaine is actually particularly hard. It's very hard to quit. But in this case, it's still true that in the first month after the intensive outpatient treatment program through the VA, the individuals that received real TMS were much more likely to remain sober. There's currently no FDA-approved treatments for cocaine. And so in the presence of no approved treatments, this is sort of a very positive sign, which we hope to continue. We haven't actually finished analyzing the brain data yet. As you can imagine, a whole lot of functional MRI data from all those people at 4 different time points, dealing with things like motion and all of that. But what I can show you is that it seems that the primary effects of the intervention are driven by the changes in medial prefrontal cortex activity to cues amongst the alcohol users in a very clean way. In the cocaine users, we get lots of changes and decreases in cue reactivity associated with the anterior insula, the inferior frontal cortex, the motor cortex. It's much sort of sloppier or less parcellated, which is interesting in general. And so kind of the punchline I wanted to highlight for you here is that medial prefrontal cortex CTBS, or I should probably say TMS to the left frontal pole, has a lot of promise, I think specifically for these alcohol users and cocaine users, and that it is actually feasible and it's well tolerated if you ramp the dose up slowly. So that's really great. And a few of our collaborators are evaluating it for different indications, including obsessive compulsive disorder. But one thing we noticed, as you often do when doing research, is there's a lot of variability. And so as we were studying these large cohorts of people in other studies, we started to realize that in fact, a lot of our alcohol users and cocaine users have pretty significant gray and white matter atrophy. And that's not new, right? I mean, I could have gone to any radiologist and they would have been like, yeah, we've known that for 30 years. But we don't usually think about that in terms of tailoring a TMS treatment to a patient. But I suspect some of you on this call probably treat maybe geriatric depression, maybe depression in people that also have been taking a lot of antipsychotics, which we know affect brain volume, heavy alcohol drinkers. So all of these things are going to change both the distance potentially change the distance from the scalp to cortex, as well as alcohol will promote cortical thinning so thinning of the cortical ribbon. And we have demonstrated before that using a technique called interleaved TMS, fMRI, you can actually show that the signal propagation from the frontal pole to the striatum is dependent upon, or is, I guess, more technically is strongly correlated with gray matter density in the area of stimulation, as well as the integrity of the white matter from that area to the terminus in the basal ganglia. We've published this in 2 different ways from 2 different cohorts, which if you're interested in, I'd love to discuss with you. I think, all of this great stuff coming out with SimNibs and people incorporating electric field models into their predictions is a really exciting way that the field is moving. And then kind of the last area I just kind of want to mention in terms of developing TMS as an emerging treatment for addiction. Those of you on the webinar that treat patients, you're probably acutely aware of the idea that addiction there's a stage, right? So usually an individual, when they start using a drug, and this is true for many drugs, they use it for the appetitive aspects, right? We pull all the way back to the first things I was talking about. It's this drive, value, the reward. But over time, it actually just becomes a habit. And that's much more of this sort of motoric aspect of it. And somewhere in there in the spectrum between emotional drive and then smoking in the morning because it's just what you've always done, is cognitive control. And so maybe we need to be smart about figuring out sort of specific biotypes or looking at the temporal emergence of activity in these individuals. And that can help us sort out which of these different TMS targets to go after for a therapeutic. And so with that, thank you all so much for your attention. Again, I wish I could be there in person with you. But to just sort of introduce you, I came from Medical University of South Carolina, where I learned to do TMS from Mark George. And we had a team of people still there at MUSC, and then we moved to Wake Forest last year, and we have this great new group there. And then I also, of course, wanted to thank you all for this invitation. I feel like I've heard about so many of you, especially the members of the executive board, for so long that it's really, it's very humbling to be invited. So thank you so much. And then just kind of last thing, as a little advertisement, we have this great new center that we're starting, and we're recruiting all kinds of people. And so if you're ever interested in coming or evaluating some type of job, or you just want to come and give a talk, we'd love to have you. Thank you so much.
Thanks so much, Colleen. A really interesting talk. Thank you for presenting. I think we can open up now for a couple of quick questions. If anyone has anything I'd like to ask Colleen, feel free to jump in and to ask. Yeah. While we're waiting, Colleen, I might just ask you a quick question. I'm curious about the dosing. It seems that you use quite substantial dosing for your studies, and it's something I've seen as well in some of the depression literature. I'm thinking about sort of the work from Nolan Williams' lab recently as well with the accelerated theta burst. Do you think that that's something that's important and that we should actually perhaps be increasing the dose with some of these things?
Yeah, that's a great question. I think that's $1 million question, really, right? But so you all have done, I guess, several people on this call have done a lot of good accelerated theta burst dosing studies and non-theta burst dosing studies. I think that certainly the idea of doing more than one session per day is very attractive, and it seems right. However, I think it's key as to like how long we should wait between each session. And in our hands, I think you could probably consider what we did one session, but it's one session of a whole lot of pulses. So two students in my lab did a recent dosing study, which is under review currently. where we did 600, 1200, 1800, and 3600 pulses of intermittent theta burst, and then the same thing with continuous theta burst. And we had 60 people in the study, 30 people received all the ITBS, 30 all the CTBS. And in general, we didn't find what we expected, and probably 1200 pulses of intermittent theta burst caused inhibition most reliably. And we had another candidate that was excitatory most reliably, but it wasn't what you thought. And so what I take away from that is, if we just think about dosing in terms of the number of pulses per session, it's not clear to me that more is better. And so in our own studies, we've actually come away from 3,600 pulses, and we're back to just using 600.
That's interesting.
That was a long answer, sorry.
But. Does anyone else have any other questions? I've just got a question in the chat here from Trevor Brown asking, why do you hold more promise for alcohol addiction as opposed to, I presume, some other addictions?
Great question. So in our hands, we've studied smokers, cocaine users, alcohol users, and opiate users with pain. And the alcohol, so especially over the forehead, I mean, it's not painless, but the alcohol users don't seem to actually experience the painfulness of TMS at all. So they can just like sit there and take it, and it really doesn't bother them. I think what we're doing now for, so we have a few big grants right now where we're still doing the frontal pole, but we're doing kind of accelerated 600 pulses, and then we wait about 30 minutes and we do a behavioral intervention in the middle, and then we do another 600 pulses. And we find the alcohol users are particularly engaged in the behavioral treatment, a little different than some of our others. So I just think those are kind of positive signs.
Interesting. Just got another question here from Sarah saying, thanks for the talk. Could you please elaborate a little more on how you measured connectivity in the scanner? Yeah.
So probably the slide that I showed was when we had the medial prefrontal cortex, and then we looked at all these other brain regions. So that was seed-based connectivity. I think we used PPI. So we used PPI, and we did it the long way. It was before the Con toolbox existed. So we just basically did kind of whole brain seed to voxel connectivity, but with these like predefined ROIs.
Okay, great.
I think James Coxon has a question.
Yeah, thanks for a really interesting talk, Colleen. I'm just wondering whether you have any ideas about whether you think that your mechanism with the stimulation protocol you're using is one of a plasticity one like LTP or a virtual lesion approach.
So that's interesting. So when I went into this, I thought the mechanism was going to be inhibiting the medial prefrontal cortex. That's why we chose CTBS, which was we thought was going to be LTD like. So that was the idea. I'm now no longer convinced that's true, just because of a lot of stuff that's come out. And what we it's probably a little too much to get into this now, but what we actually find is that although functional connectivity goes down, in many cases, bold signal at the site remains the same or potentially even increases. So it's as though we can disrupt kind of the correlation or the synchronicity perhaps of these frontal striatal terminals. But in fact, we seem to either not really be affecting bold signal in the region we're stimulating, or we're increasing it with 3,600. So I don't really think it's like a lesion, but it is blocking signal transduction or something like that.
And I think we've probably just one more question we've got here from Claire Bradley saying, thanks for a fantastic talk. Just a note on maintenance sessions that you mentioned, some colleagues of mine in France treating neuropathic pain for patients have noticed repeated single sessions of 20 Hertz RTMS in the motor cortex, spaced a couple of weeks, give a larger benefit than sham and can be used to maintain pain relief. Would love to hear from you and others if you've noticed anything similar in your patients.
Yes. Yeah, and you're in France. I feel like I don't know how to properly pronounce the name of the lead author on all of those papers, and I'm sure I'll butcher it. But yes, you all are actually the kings of TMS as an analgesic treatment. Yes, we're exploring, I have not done 20 Hertz, but we have a grant right now where we're doing ITBS to the motor cortex versus the dorsolateral versus the ventral medial for pain. And oh yeah, the motor cortex is the best in our hands for sure. Even in like promoting opiate sparing. That's great. Yeah, I'd be happy to offline if you're interested.
All right, I think probably in the interest of time, we might just leave it there. But thank you again, Colleen, for a really interesting talk, and for joining us from the other side of the ocean as well. Appreciate it.
Thank you all so much.